Alright, so let's talk about meiosis. Meiosis is the process that produces a sex cell, or a gamete. Y'all ever heard of a sperm? That's the male gamete. An egg is the female gamete. Each gamete only contains one copy of each chromosome, and this is why when you have two organisms that mate, each pass down only one copy of their genetic information. So anyway, this process to create the sex cells, the gametes, how does that happen? So you have a cell, and this is a normal cell, and the cell has a nucleus. Basically, inside the nucleus, and we're only going to follow one chromosome. There's going to be a ton of chromosomes, we're going to just look at one. There's a chromosome, and there's two copies of it. It's diploid, which means that there's two copies of each chromosome. Di meaning two, and ploid, like ploidy, referring to the number of chromosomes. And these chromosomes are not condensed. They do not look like how you think of a standard chromosome, which is like this X thing, you know? You've probably seen that in a karyotype, or an image that shows all of the chromosomes like lined up. And basically, this cell is in interphase. When a cell begins to want to divide, the first thing it does is it duplicates each chromosome. So now there's a copy of each chromosome. And when I said there's basically two copies of each chromosome, here, diploid, they're different. They have different alleles. This has one allele, this has another. But here, these are exact copies of the two that we started originally. So it replicates its DNA. And this is also a part of interphase, and we'll look at the complete map of the cell, basically the cell cycle at the end. And it's still not condensed. Now, the cell's like, okay, I have enough DNA to basically split myself. We start prophase. We start Prophase I. Basically, there's a couple of things that happen, and I'll draw them kind of separately. These little protein dudes, I'm going to draw them like this, called centrosomes. They start to extend to these microtubules, which are kind of like these structural proteins, and they extend from them, so we can kind of draw them like this. And the chromosomes also condense and form the standard X that we're used to, and the chromosomes pair up. So these two chromosomes, they pair up, they find each other. And the last part is that the nuclear membrane dissolves. Alright, so let's review the steps of Prophase I. Centrosomes extend microtubules. Chromosomes condense. Chromosomes pair up. Nuclear membrane dissolves. Awesome. And now we're going to go on to the next step, Metaphase I. Metaphase is kind of everything getting in order. Prophase was kind of setting the stage. Metaphase is like getting everything in its correct place, and then the later stages take it from there. So you can kind of think of a cell as a planet. Let's say that like the North Pole is up here, and the South Pole is up here, and then here's the equator. We would have one centrosome up here, and the other down here. They have migrated to opposite poles, and then all of the chromosome pairs would align themselves basically along the equator. And normally we draw it kind of looking like this, with the centrosomes on the left and the right, and then the chromosome pairs kind of up and down the center. So like we've only been following one pair, but there would be others up here and then others down here. So the chromosomes line up along the equator of the cell. And now remember that these centrosome dudes have these microtubules that they've extended. And these are going to extend to all of the chromosomes. And as you can see, each of them, like this guy takes all the ones on the left, this guy takes all the ones on the right. This is called the meiotic spindle. And the meiotic spindle binds to the centromeres of the chromosomes. We look at a chromosome like here's one chromatid, and it's attached to the other at a point. This is called the centromere, and the meiotic spindle binds to the centromeres of the chromosomes. So let's review that. Centrosomes go to opposite poles. Chromosomes align along the equator. Meiotic spindle attaches to centromeres. Alright, so basically everything is set, and you can probably tell that these guys are going to pull the chromosome pairs apart. This is Anaphase I. Anaphase I is where the meiotic spindle retracts. They literally separate the two chromosomes to the opposite sides of the cell. One is over here and the other is over here with these dudes, you know. The meiotic spindle pulls the chromosomes apart from each other to opposite poles. Next, we have Telophase I. We've done the work of separating the chromosomes. And so now what happens, that nuclear membrane reforms around each of these chromosomes. Remember, it dissolved up in Prophase I. And now that we have separated these two copies of the chromosome, we reform the nuclear membrane. So you can see it's pretty much ready for two new cells to be formed. And another thing is that the DNA decondenses. It just relaxes. So now it kind of looks like this. The next part part is Cytokinesis I. This is the final part of Meiosis I. The cell, which has 2 nuclei, pinches in the center and divides. Awesome, so now we have 2 cells, and each of these cells has 2 copies of one version of the chromosome. Now we can go to Meiosis II, the second part. The first part was Meiosis I. We're going to repeat pretty much everything. Let's go to Prophase II, and this happens to both of the cells that were produced in Meiosis I, but we're going to just focus on one of them. So now we have a cell, and the chromosome is not condensed. So what we're going to do in Prophase I is condense the DNA. It turns back into that X. In each cell as well, a centrosome hung along, because these guys were kind of like chilling at the edges of the cell, right? They got separated, went to opposite poles, and this guy's going to duplicate himself, and they're going to start to form their microtubules. And then finally, the nuclear membrane is going to dissolve once more. So let's review that. DNA condenses, centrosome duplicates, I think it's spelled like this, microtubules extend, and nuclear membrane dissolves. Awesome, we've done the first step of Meiosis II. Now on to the second step, Metaphase II. Remember, prophase is basically getting everything ready. Metaphase is getting it to place. So the centrosomes are going to go to the opposite sides, opposite poles, and the chromosomes are going to align along the equator of the cell. And then the meiotic spindle, the microtubules, would attach to the centromeres. Alright, centrosomes go to opposite poles. Chromosomes align on equator. And the meiotic spindle attaches to the centromeres. Perfect. Everything is in its place. So now we have Anaphase II. As you can see, this is pretty similar to the first round. First we were separating these guys, and now we're pulling each one of those apart. Pretty much the exact same thing happens. They pull them apart. The meiotic spindle pulls the sister chromatids to opposite poles. Alright, now we're at Telophase II. You probably guessed it. Nuclear membrane reforms, each one of them containing just this one little dude. And an interesting thing about chromosomes is that it kind of works like lasagna. Like, if you have a lasagna, and here's my drawing, and you cut it in half, you now have 2 lasagnas. And then if you stack them back on top of each other, you have one lasagna again. And the exact same thing happens with chromosomes. If you have basically these 2 dudes joined together, that's one chromosome, and they're called sister chromatids. But if you separate them, they now become their own individual chromosomes, and we call them chromosomes. And they also relax. The nuclear membrane reforms, chromosomes relax. And the final step, we'll see if I can fit it in here, is Cytokinesis II, where the cell pinches in the middle and divides. So let's look at kind of like the entire process. Sometimes you will see like a wheel for the cell cycle. And we have the S phase, G2 phase, the G1 phase, and then meiosis. So basically G1 is growth. And let's say that a cell life starts here. It has just been formed. So it's growing. And then it hits S phase, which is DNA replication, or synthesis. It keeps going and then hits G2 phase, where it's another growth, preparing for meiosis. And then this is meiosis. And then within meiosis is the prophases, and the metaphases, and the anaphases, and the telophases, and everything we just discussed. So as you can see, it's actually not the majority of the cell life. Most of the time, it's preparing for this. Oh, and all of this is interphase. And then within here is everything else. So yeah, let's just summarize. Interphase, growing or replicating DNA. Prophase is basically getting all of the things ready. So it's DNA condensing, chromosomes pairing up. Metaphase is where everything gets into place. Chromosomes on equator, centrosomes on opposite poles, meiotic spindle attaching to centromeres. Anaphase, pulling chromosomes apart. Telophase, DNA relaxes, the nuclear membrane reforms. And cytokinesis, where the cell splits. And each of this happens twice, in Meiosis I and Meiosis II, where Meiosis I separates the pairs of replicated chromatids, where in Meiosis II, it separates the replicant sister chromatids from each other. And that's about it.